Study hard until you can say, trust me, I am your doctor. Hi, all neat aspirants, those who are at the gateway to the doctors. Count down 15 days for last minute preparation. We, Darsana Academy, with 3 decades of experience, managed by CMA fathers, here before you. Quick revision of plus one botany through previous year question discussion. We are going to begin with living world. While go through the previous questions, we can see that in 2019, 18, and 16, there are one question asked. We know that this chapter is simple chapter, but this chapter is connected to second year first chapter, reproduction in organism, and also morphology of flowering plants. We can see that which are the different points in this chapter. Now we know that while we discuss the concept, there will be more time. Therefore, I just give the name of the areas. Then we can give more details about that. Okay, we can go through that. You just thoroughly prepare all these things points. And what's the point? Characteristics. What is characteristic? We know that there's a defining, non-defining. Study that point. Very important. Next, we can see the taxonomy. Which are the steps of taxonomy? That's important. You have to be study that. And taxonomy categories. Which are the taxonomy categories and what is taxonomy? Very point, nook and corner, you have to be read and study carefully. That's very important. And next we can see that taxonomical aids, which are the different taxonomical aids, and very important point this chapter, and also systematic is also there in this chapter. And while go through these questions of this chapter, we can see that what's the first question? Study the 4 statements given below and select the 2 correct ones out of them. These are pattern of question. Prepare these type questions also. And what's the point? We can see that. First option, definition of biological species was given by Ernest Mayr. What about Ernest Mayr? We can see that while discussing questions, people give more weightage to the concept and we can memorize, we can recollect all the points in short way. That's the way we are going to prepare today. Okay, and we can see that. What about Ernest Mayr? We know that he, it was a point, different point related to Ernest Mayr. We have to be noticed before going to exam. We know that he is considered to be Darwin of 20th century, very important. Next one, we can see that currently accepted definition of species by Charles Darwin. And also he had awarded with Triple Crown of Biology. That's also important. Next, we can see that second. Yes, what's the second option? Photoperiod does not affect reproduction in plants. We know that this photoperiodism is connected with last chapter of botany, that is plant growth and development. We know that duration of light period affect that is growth of plant, especially flowering. That is called photoperiodism. We know that there is long day plant and short day plant. The long day plant is summer flowering plant, and short day plant is winter flowering plant, and day noted plants also. But these plants affect light. That is that for the flowering. These that depend. That is here does not affect it. That is wrong. Absolutely wrong. We know that. And what's the third one? Yes. Binomial nomenclature system was given by R.H. Whittaker. What about R.H. Whittaker? We know that he is considered to be, he is proposed 5 kingdom classification in 1969. Every year in the NCRT, you have to be studied thoroughly, make tips for that, make code for that, and that's very important. And what about binomial nomenclature? We know that Carolus Linnaeus binomial nomenclature, okay? And next question, next option. In unicellular organism, reproduction is synonymous with growth. You know what is about growth? We can see that growth that is connected with the chapter. A cell cycle that connect. That is the point is mitosis, and that is in the unicellular organism. We can see the cell division and also reproduction. That is by mitosis. Yes, that is correct. And what is the question? What is this question? The 2 correct statements. These type of questions are there. The 2 correct statements are, which are the correct statements? We can see that to the correct statements, and we can see what are the options. We can see that yes, A and D are the correct options. We know that, and that is the correct answer. Thorough go through these options. They're very important. Carefully read all options. That's important. And we can see that A and B and C, B and C. And next we can see that C and D. And what is the correct answer? Yes. What's the correct answer? Yes. 1 is the correct answer. Okay. And what about the next question? Select The correctly written scientific name of mango, yes, which was first described by Carlos Linnaeus. And again, Carlos Linnaeus. Yes, important man for the entrance. We know that. Yes. Father of taxonomy? Yes. Binomial nomenclature proposed by Carlos Linnaeus. And S, species planner is very important contribution, and system and natural. These are the points related to Carlos Linnaeus for Neet exam. And we know that. What about taxonomy categories? This mango, that is scientific name, that is binomial nomenclature. We know that which are the taxonomy categories, which are they? That is species, genus, family, order, class, division, or phylum, and kingdom. And while go through these taxonomy categories, we can see that more similarity can be seen in lowest category species, and higher similarity can be seen in kingdom, and more members come under kingdom, least members come under species, and similarity will be decreased from lower to higher category. These type of points are there. And one more point we can correct, each one is taxon. That is unit of classification is taxon, group or category. That's important. Okay. And next we can see that what's the option for this question? What's the question? Select correctly written scientific name. That is not the correct written scientific name is we know that Mangifera indica, that is correct. But that's a complete question, which was first described by Carlos Linnaeus. Is described. That question is also, that type of question also there. But in this case, question is like that. Answer is also there for the exam like that. We can see that what is the answer? Mangifera indica Linn is the correct answer. We know that. And this is Mangifera indica. This is a correct written name, but that is not description also come this first option. And what about third, Mangifera indica? Here we can see what is the written name, what is the wrong in this statement, in this Mangifera indica, I capital letter. What about the binomial nomenclature? We know that this name consists two components. First one is genus, Mangifera is genus, and indica is species. And it is, we know that that is when handwritten, separately underlined, and printed in italics. That was also a question for the entrance exam. Okay, and we can see that Mangifera indica Carlin, that is wrong. What's the correct answer? We can see that. What's the correct answer for this question? Is one. This is the correct answer. Do like this. While you see any option, just go through that and how you can connect. This time you just use like that. Then you can improve more in biology. We know that out of total 180, that is 180, 90 questions of biology. You have to be scored full marks biology. Then you read carefully all options. That's very important. Avoid careless mistake. That's also very important. Concept clarity is important. Connection have to be maintained. That's also important. Okay, indirect question may be there hidden. We can see this type of questions while we discuss. Okay, and what about the third question? Merge the items. That type of questions are there. Merge the column is also there. Merge the items given in column 1 with those in column 2 and select the correct option given below column 1 and column 2. Yes. We can see that. What about herbarium? We can see that it is a place where dried and pressed plant specimens mounted on sheets are kept. That is arranged based on accepted system of classification. And this herbarium sheet carrying label, that is also important. Information such as that is English, local, scientific name of plant, family of the plant, then collector's name, date and place of collection. That is also a question for the entrance exam. We know that. And what about next? This herbarium is the quick referral system for taxonomic studies. That is also for the question. That is also a question for the entrance. We know that. And next we can see that key. What about key? A booklet containing a list of characters and their alternates, which are helpful in identification of various taxa. That is alternate characters. That is called dissimilar pair of statement is called. What's answer? Is couplet. And each statement is called lead. That's also correct answer. That's also question. And what's the bond related to key? We can see that it is an analytical device. That type of question is also there. Okay. And what about the third one? Museum. Yes, what is museum? We can see that it's a place having a collection of preserved plants and animals. Not only plants and animals, and also that is can not only this preservation in preservative solution, and also skeleton of animals also there, and mammals and birds can be stuffed, and also insect can be collected in insect box. These type of questions also there. And what about next one? Catalog. That word can be seen in the NCERT. That is NCERT based questions. That meaning you have to go through every word, single word in the NCERT for the entrance exam. We can see that. Yes, a list that enumerates methodically all the species found in an area with brief description aiding identification. Yes, that is catalog. From here, you can see that. What's the correct answer? Yes, Carefully read all the match and then match the column. Also read an option also very important. And what's the correct answer for this question? We can see that 4. Yes, 4 is the correct answer for this question. And next we can see that this is herbarium sheet. Yes, there is the labeling is also there. Plants dried and pressed on the herbarium sheet. And we can see that this is key. That is types of leaf, that is simple and compound, that is pair of alternative or dissimilar pair of statement. This is couplet. One will be select. If leaf is simple, we can select that. And then go to the next page, 181. That's the key. And next we can see that this is museum. Yes, collection. We can see that here. What we can see, the skeleton are there, preservative solutions are there, mom is stuffed. Yes, that's the point. Next we can see that. What about the next chapter? Yes, biological classification. This chapter is also very important. While go through the previous questions, we can see that. Yes, in 2019, that is 2 questions, whereas in 2018, 17, 16, 15, 4 questions asked. Very important chapter. And there are certain concepts and examples are there. Study all the points. We can see what are the different area in this chapter. There is 5 kingdom classification. Already we know that that is proposed by Robert Harding Whittaker in 1969 and also considered which are the characters considered also important. And we can see that which are the 5 kingdom. Monera. Yes. They are. That is prokaryote kingdom. That is its all member is bacteria. That bacteria is connected with, that is prokaryotes is connected with cell, the unit of life. While we go through the prokaryote cell, that we studied this kingdom Monera. We know that there are 2 types of bacteria, archaebacteria and eubacteria. Yes, eubacteria are of 3 types: photosynthetic autotrophic bacteria, chemosynthetic autotrophic bacteria, and heterotrophic. Bacteria is the abundant form of bacteria. This connects. Okay. Next, we can see that protista. Yes, what about protista? Is a unicellular eukaryotes. Chrysophytes, dinoflagellate, euglenoids, slime mold, protozoans are the classes of kingdom protista. We know that. And what about kingdom fungi? Very important question for the entrance. Multicellular, eukaryotes, heterotrophs with, yes, cell wall. Yes, that's correct. Next, we can see that kingdom plantae. What about the points? Yes, multicellular, eukaryotes, heterotrophs, autotrophs. Plantae are autotrophs. What is the difference between fungi and plantae? Fungi is heterotrophs, whereas plantae is autotrophs, but they have cell wall. Both have cell wall, but what's the difference? These the fungi are heterotrophs, whereas plantae are autotrophs. We know that. And what about kingdom Animalia? Multicellular, eukaryotes, heterotrophs without cell wall. With a cell wall, fungi. Without cell wall, Animalia. Yes. These type of points are there. Cunning preparation you have to be keep during these days. Be confident. Okay. And we can see that certain points, other than 5 kingdom classification virus, certain scientist name are there, years are there, very important years are there, and certain examples are there. Study all the points. You can corner, read and study. Okay, and next we can see that viroid, there is no protein code. Okay, the only RNA, that is teodyner in 19. Yes, what's the answer? 1971. Yes, memorize. Singer Nicholson in 1972, but here it is 1971. Yes, that is prion. It's proteinaceous infectious agent. That's called prions. There's mad cow disease, BSE, bovine spongiform encephalopathy. Yes, CJD, Creutzfeldt Jakob disease. Very important point. Later to prions. Study all the points. Okay. And next we can see that what's the question? Select. Wrong statement. Yes. What's the point? Cell wall is present in members of fungi and plantae. Already we had seen that they have cell wall. Fungi have chitin cell wall. Plantae, we know that cellulose cell wall. In the case of parenchyma, collenchyma, cellulose, hemicellulose, pectin, whereas sclerenchyma has. Now we know that lignin. Yes, study like that. And we can see that. What about other members with cell wall? They are bacteria. Peptidoglycan cell wall have bacteria. And what about others? Protistants are also the which members? Chrysophytes, diatom have silica cell wall. And dinoflagellate have they also have cellulosic cell plate. This dinoflagellate. Yes. And connect, no? We just see the option like that is cell wall is present. But which are the members those have cell wall? Just think about that. Then your thinking skill and your connection skill can improve during these days. Okay. And next we can see that. Second statement, second option. Mushroom belongs to basidiomycetes. What is mushroom? We can see that they are. Other members, they are agaricus. Other members of Basidiomyces, we can see that. Study all the points, bracket fungi. Then we know that puff boars. And then we can see that Eustilago. What is Eustilago? That is smut fungi. Very important point. And then we can see that Puxine is rust fungi. Study all the classes and examples and all the points. Okay, and then we can see that, yes, that's correct. Next one, we can see that what is the third option? Pseudopodia, locomotory, and feeding structure in sporozoans. What about sporozoan? Sporozoans are protozoans. Based on locomotory organ, there are four types. Amoeboid protozoan, move the pseudopodia, not sporozoan. Flagellated protozoan, ciliated protozoan, and sporozoan. What about sporozoan? They are protozoan and they have no locomotory organ. What's the example? We can see that Plasmodium vivax, cause malarial parasites. It's a malarial parasite. And come under kingdom, Protista. We know that. Okay. And what is about here? We can see third is wrong. We can see that. Next is option. Mitochondria, the powerhouse of the cell in all kingdom except Monera. What is the specialty of Monera? We know that Monerans are prokaryotes. What is prokaryote? They have no true nucleus and they have no membrane-bound organelle, whereas mitochondria is, we know that these prokaryotes have no membrane-bound organelle. And what about this mitochondria? That is in eukaryotes. What's the specialty of mitochondria? That is in eukaryotes, they have membrane-bound organelles. We can see that have two membrane in the case of mitochondria. Just think about which are the organelles, those have two membrane. We can see that which have they? They are chloroplast. And also nucleus, that point also there. And we can connect this chloroplast and mitochondria like, what is the point? Yes, they are semi-autonomous organelle also. This type of connection also you have to be developed. And we can see that what's the answer for this question? Yes, 3 is the correct answer for this question. Yes. Okay. And what about the next question? After karyogymy, followed by meiosis, spores produce exogenously. And there are no two concepts are there in the question. Very wonderful question, beautiful question. After karyogamy followed by meiosis, we can see that after karyogamy followed by meiosis can be seen and there is karyogamy fusion of. Yes, we can see that what is karyogamy, plasmogamy, karyogamy is fusion of nucleus. Zygote will be formed. Zygote, after zygote formation, meiosis takes place in algae, those have haplontic life cycle, and also fungi. But spores are produced exogenous. That's a very wonderful question. In this case, fungi, we can see that there are spores are produced exogenously in one member. We can see that. What's the option? Yes, what's the option? Neurospora. What is neurospora? Yes, what about neurospora? That is ascomyces, that is sac fungi. Okay, in this case, we can see that ascospore are produced, that is sexual spore, that is produced endogenously, that is not exogenously. Yes. And conidae, asexual reproduction in ascomyces, they are produced exogenous. That type of questions were for the entrance. Okay, and therefore we can see that that is not the answer for this question. And next we can see that. Second option, alternaria. What about alternaria? Who is alternaria? Yes, we can say that they are deuteromyces. What about the specialty of deuteromyces? We can say that they are imperfect fungi. What is imperfect? There is no sexual reproduction. Yes. And what about which are the perfect fungi? Ascomyces, basidiomyces, and also phycomyces are the perfect fungi. And we can see that other members of this class deuteromyces are trichoderma, yes, and then Galactotrichum, example very important. Okay, and that is not the answer. And next we can see that agaricus. And what about agaricus? Agaricus is mushroom, and we can see that they are basidiomyces. Same concept in different angle questions are there. And we can see that basidiomyces, and what is the specialty of basidiomyces? They have basidiospore, that is sexual spore produced exogenously. That's very important. Sexual spore produced exogenously. That's important. Okay, and we can see that there is no sex organ. In the case of basidiomyces, there is no sex organ. That's important. Sex organs are not there. Very important question for the entrance. And asexual reproduction are generally not that sentence in the NCERT. NCERT sentence pakka will be asked for the entrance. Okay, and next we can see that. What about the last? Saccaro. What is the exogenous produce? We can see the agaricus, of course. Instead of agaricus, if option like, then what are the different example? Puffball, Ustilago, Paxin? Yes, that's answer. Yes, based on NCRT, that will be the answer. And what about Saccharomyces? Saccharomyces, what is Saccharomyces cerevisiae? Yes, that is yeast. Very important question. Yeast have no mycelium. Very important. They are unicellular. Yes, we know that. And they are come under ascomycetes. Ascomycetes. And we know that they are known as that connected with microbes in human welfare. That is brewers and workers yeast. And also we can see that Saccharum officinarum. Are you familiar with that? Saccharum officinarum. Yes, that is in strategies for enhancement in food production. We can see that they are sugarcane. But this is not Saccharomyces officinalis, this is Saccharomyces. This type of careless and fast hurry will be in the exam hall. Avoid that. Then you can become a doctor, there is no doubt. We Darsana Academy here for you, right? And what is the answer for this question? We can see that option 3 is the correct answer for this question. And what about next question? Which among the following is not a prokaryote? Not a prokaryote means if at the time of during exam, no, maybe you feel tension and you just read like is prokaryote, then it's over, right? Is not prokaryote. Carefully read and underline that, then you can write the answer. What's the option? We can see that. First option is Saccharomyces. What about Saccharomyces? Saccharomyces cerevisiae is yeast. Yeast. Yeast is fungi. Fungi is eukaryote. Yes, that is not prokaryote. Option is right. This is right option. And what we can see, and this is fungi, and they are eukaryotes. Already we had seen that. They come under ascomyces. And they are this, what about the yeast? They are unicellular organs reproduced by budding, asexual reproduction. That's also a very important question for the entrance. Okay, and next we can see that Mycobacterium is the second option. What is Mycobacterium? Of course, that is a genus of actinobacteria. They are bacteria. Bacteria are prokaryotes. And we can see that they come under kingdom Monera. Mycobacterium tuberculosis causes tuberculosis. Okay, of course they are not prokaryote means they are prokaryote. They are not eukaryote means they are prokaryote. Yes, question is not prokaryote. Okay, and next we can see that what's the next question? Option. What is no option? Nostoc. What about nostoc? You're familiar with nostoc. And what about nostoc? They are eubacteria. Okay, they are true bacteria. They are cyanobacteria or photosynthetic autotrophic bacteria. We know that. And what about this nostoc with anabina? Nostoc and anabina. Nostoc with anthoceros. Anabina with azola for symbiotic nitrogen fixation. Nostoc and anabina. Very important examples from cyanobacteria. Very important. And we know that they are also called blue-green algae. And next point we can see that oscillatoria. What is oscillatoria? We can see that they are eubacteria. They are true bacteria, oscillatoria. And we can see that they are cyanobacteria, they are photosynthetic autotrophic bacteria. But this oscillatoria term we can see in the chapter microbes in human welfare that used as biofertilizer. Okay, from that, this question asks, while we are in the chapter, this chapter biology classification, but the question is also from here and there. From here and there, the option. Therefore, in a single question consists many chapters. That is the importance. You have to become a connection, scratch from here and there and prepare well. That's very, very important during these days. Okay, and we can see that. That's about this chapter, and we are going to discuss next chapter. Yes, what is the answer for this question? Already we had said that one is the correct answer. And what about the next chapter, plant kingdom? While go through these previous questions of this chapter, we can see that in 2018-17 there are 4 questions asked, whereas in 2019 and 16 there are 2 questions asked, and whereas in 2015 also there is 4 questions asked. Very important chapter. In this chapter is also there is examples, diagram, and certain diplonic, this concept carefully study. Very interesting chapter, this plant kingdom also, we know that. And what are the different concepts in this chapter? We can see that there are system of classification, natural system, artificial system, and phylogenetic, numerical taxonomy, cytotaxonomy, chemotaxonomy, sentence in the NCRT, some words in the NCRT important for the entrance in this area. And we can see that next, algae, divisions of kingdom plant, algae. What is about algae? Algae is haplontic life cycle. But algae with diplodoctic life cycle is fucus, and haplodiplodoctic life cycle is Polysiphonia ectocarpus kelps. Important question for the entrance. And we can see that bryophyte. What about bryophyte? Plant body is gametophyte. They have haplodiplodic life cycle. And what about pteridophyte? The plant body is sporophyte. They also have haplodiplodic life cycle. And what about the gymnosperm? There, this plant body is sporophyte, and they have diplodic life cycle. And which is the most evolved one? We can see that they are angiosperm. Angiosperms, the plant body is also sporophyte, and they have diplodic life cycle. And these are not the points, and you have to be study thoroughly all the points in the NCERT and make code and make connection. That's important. And while go through the question, we can discuss that in concept also. Okay, and we can see that. What the question? From evolutionary point of view, retention of the female gametophyte with developing young embryo on the parent sporophyte for some time, notice for some time, is first observed in, that's the question. The question contains a certain point. You think that biology is important, easy, is very important, we know that, but it is easy, yes, it is easy, but those who are prepared systematic way and those who have maintained connect with one chapter to another chapter, then your question will be easy for you. There's no doubt. Okay, we can see that that is for some time is first observed in. What's the answer? Options. What about the option one? Mosses. What is mosses? We can see that they are bryophyte. Mosses are bryophyte. Okay, and they have the plant body of mosses have protonema and leafy stage. Important. They are gametophyte, the plant body, we know that. Haploid. In the case of mosses, we know that they are homospores, that is, produce one type of spores. Therefore, they have no this type of this developing embryo never attached on the parent sporophyte. Those are heterospory, they can. Okay, and what about the second option, pteridophyte? And what is the specialty of pteridophyte? We can see that generally they are homosporous, of course. They are homosporous, but heterosporous members are there. Very important point for the entrance. That's also question asked. You know that. Salvinia selaginella, very important question for the entrance exam. And we can see that since this heterospory, this is considered to be precursor of seed habits. Precursor of seed habit is very important question for the entrance exam. And we can see that in this case, developing embryo on the parent sporophyte, that is attached, female gametophyte with developing embryo on this parent sporophyte for some time will be attached. In the case of what's the answer for this question? Pteridophyte is the correct answer for this question. And what about the third option? Gymnosperms. What about gymnosperms? Of course, they are heterosporous. But what's the point? Developing embryo on the parent sporophyte, that is always there. That is not for the sometime in the case of gymnosperms. And also ovules are not enclosed, that is naked. Gymnosperms, sperm, seeds are naked. We know that. And branch is cycles, very important. And branch is also important. Pinus, citrus, that's also important. Okay, these are the different questions connected to different points. You can develop this skill and during these days, go through the questions, select option and make points, and then you can develop your memory skill like network, like spider network. You can memorize, you can develop your memory. That's important way for getting full marks for biology, and your sharpness will be increased. There is no doubt. And next we can see that option 4 is liverworts. And they are bryophyte. Liverworts are bryophyte, we know that. And they are know this there. That example is Markantia. Markantia, very important. And we can see that this Markantia with Archegonia 4, Antheridia 4, that can be seen in this plant kingdom and also in reproduction in organism. This connection can be seen with one chapter to another chapter. That is very important. You just notice all these points. And for this question, what's the answer? We can see that what's the answer for this question? We can see that second option is the correct answer. Pteridophyte is the correct answer for this question. Okay? And we can move to the next question. Which one is wrongly matched? Very important pattern of the entrance. Wrong match or mismatch, correct match, true match or true combination, like that. Important question for the entrance. And we can see that, first one, uniflagellate gamete polysiphonia. You just think about polysiphonia. Who is that? Polysiphonia. We know that that is rhodophyceae, that is red algae. They have no flagella. We know that. Yes. And what are the other members? If instead of Polycephalus, if it is there, Porphyra, Gracilaria, Gelidia. Yes. Members very important. Examples very important. Examples which are given in the NCERT, that is important. Example which is given in the NCERT, that is important for the entrance. There is no doubt. Okay. And next we can see that this is Polysiphonia. Red algae. Yes, red algae. Red color. Yes. Next we can see that biflagellate zoospore. Biflagellate zoospore, that is brown algae. Yes. In the case of this brown algae, phaeophytes, we can see that two unequally. Is it equally? No. Two unequally. That point is important. Laterally attached. Yes, lateral. Is it apical? No. Laterally attached flagella. What is the shape? Pear-shaped zoospore. Very important question. Very important point for the entrance. Pear-shaped zoospore. Pyriform shape. Very important. Okay, we can see that. In this, we can see wrong is, of course, one is the correct, one is the wrong. That is the correct answer for this question. We can see that. Yes. What's the answer for this question? Yes. This is the answer. And what are the different options? What are the three and four, third, three and fourth option? We can see that. And what about the third option? Gemme kappa markantia. What about markantia? Very important. Markantia. We can. What about gemme? It's very important. What about gemme? Green. What's very important point for that? That's green. Yes. Multicellular. Is it unicellular? No. Multicellular? Yes. Asexual bud? Of course, it is asexual bud. That is written in the NCERT. That was also for the entrance question. And we can see that this is gemme on the receptacle, gemme cup. That can be seen in Markantia. They are liverworts. Okay. And next we can see that what's the fourth option number 4? Unicellular organism chlorella. Yes, of course. How can connect this chlorella? We can see that unicellular, they are unicellular algae and they are rich in protein. Yes, they are rich in protein. And also we can see that that is connected with strategies, SCP, strategies for enhancement, single cell protein, yes. And the space traveler used by, the question is also like that. Name an algae used by space traveler, that is chlorella. Yes, and also this is chlorella, right? And we can see that spirulina is also connected with space traveler. The cyanobacteria used by space traveler is spirulina. That's also a question for the entrance exam. Okay, well, go through the options. We can see all these things. Okay, and what about the next question? Question number 9. An example of colonial algae, colonial, that's unicellular, filamentous, and these type of these points are there in the NCERT. What is colonial algae? We can see this chlorella. We had seen that there is unicellular. There, unis, we had seen the previous question. And what about the second, volvox? What about volvox? We can see that volvox. Volvox is, yes, they are colonial. They are colonial. Volvox is called, what is the another point of volvox? That is oogamous. Oogamous. And fucus, that is oogamous. Fucus is also oogamous. And volvox is colonial also. Very important. And this is, we can see that option number 3, Eulothrix. Eulothrix is, we know that they are filamentous. And we can Spirogyra filamentous. And Ectocarpus filamentous. Ectocarpus branch filamentous. Very important. And we can see that Spirogyra is option. There is another option here also we discussed. That is also filamentous. In this question, we can see that what's the correct answer? Yes, we get this is Spirogyra diagram. And we can see that what's the correct answer? Yes, option 2 is the correct answer for this question. Okay. And next we can see that. What's the next question? Wind pollen grains are present in. That's important question. That was the question for the entrance. That is connected with gymnosperm member. Okay, gymnosperm. While go through the gymnosperm, certain points also you have to be connect. And we can see that. Yes, we can see that mustard. What about mustard? What's very important point in the chapter strategies and also morphology of flowering plants? We can see this mustard. No, question is from connected to one chapter and options from different chapters. Yes, that's a way. And we can see that angiosperm, mustards are angiosperms. And we can see that they are come under Brassicaceae. We can see that Brassicaceae floral diagram and floral formula intensity very important. We can see that this one. From this floral diagram and floral formula, maximum points you have to be collect. And we can see that there is actinomorphic, bisexual, calyx 2 plus 2, corolla 4, and androecium 2 plus 4, didynamous stamen, and gynoecium, carpel 2, fused, hypogynous, very important. And this diagram also very, very important. Okay. And next we can see that. Second option, cycas. And what about cycas? We can see that the very important point related to cycas is correlated roots. Cycas CCC cyanobacteria. Cycas correlated roots cyanobacteria. For nitrogen fixation, very important question. And we can see that they are cycas are unisexual, that is connected with gymnosperm. Cycas are unisexual, and this is correlated root association. Okay, very important question for the entrance. That other question, okay. And next we can see that another option is mango. And what about mango? Mango is connected with, we can see that there are anacardiaceae that connected with living world. Anacardiaceae, that is order is sapindales, very important. And we can see that is mesocarp of this mango is used to make juice, that is edible portion, important. And then we can, they are droop, mango and coconut are droop, important. And next we can see that, what's another option? Pinus. Yes. What about pinus? What's the question? Winged pollen grains are present in the case of pinus. What about pinus? Extended outer exine on two lateral sides to form two wings of pollen. Yes, other genera of Pinaceae also like that. And then we can see that how two bladder-like wings they have. And also this is the structure. Very important. What's the answer for this question? Pinus is the answer for this question. We can see that option 4 is the correct answer for this question. Okay, and then we can see that. And we already started our repeater batch, and we will start after Neet exam also, another batch also started. Okay, and we are always here with you. Okay, and next we can see that. Next question, next chapter. That is morphology of flowering plants, very important chapter, we know that. Okay, while go through the previous questions, we can see that. What is the pattern of question? No, during 2016 and 15, there are 5 questions asked from this chapter. And we can see that in 2019, 1 question, and 2018, 2 questions, and 2017 also 2 questions asked from this area. Very important chapter for the entrance exam. Okay, and we can see which are the different points in this chapter. Root and its modification. Okay, what is the NCERT given, and also based on NCERT, you also have to be prepared. Preparation level, you have to be improved like that. That's important. And then we can see that stem and modification is also there. Stem and its modification is also there. And then we can see that the leaf and its modification, study all the points. We know that while we are going to discuss these points, we don't have enough time, all these discussion. Okay, we know that. Therefore, you just go through all these points once more. Okay, make short note, make easy method to memorize these examples. That's important. Already you have to make enjoying chord and beauty chords, and you can memorize all these things. Okay, and then we can see that venation type, venation, reticulate, parallel types, that is simple and compound, and phyllotaxy, opposite, alternate, and world, and that is inflorescence, racemose, cymose, and we can see that other inflorescence also you have to be study. Okay, that's important. And next we can see that symmetry is also there, actinomorphic, zygomorphic, and asymmetric. Yes, and position of floral parts, hypogynous, epigynous, perigynous, and we can see that placentation, different placentation. We will deal this certain area while discuss the questions. And we can see that their fruits and seeds are there, and different families. There is Fabaceae, Solanaceae, Liliaceae, and representation of Brassicaceae. Already we had seen. And we can see which are the questions. Placentation, in which ovules develop on the inner wall of the ovary or in peripheral part, is. You can see that this is the same sentence in the NCERT. Based on the sentence, the question asked. Or maybe the diagram, the question will be, from the diagram, question will be asked. Or from the option, identify the diagram. These type of question will be there. From the simple question, from the same question, different level of pattern will be there for the entrance exam. That is a specialty of biology. And we can see that here, axial placentation. What about axial? What's the first option is actually axial. And what about axial? Ovules at the axis. And they have septa. Axial placentation is the character features of Solanaceae, Liliaceae. And also we can see that hibiscus, very important, and citrus, yes. And we can see that parietal placentation, that's the point. That is placentation in which ovule develops on the inner wall of the ovary or peripheral part is parietal is answer. What is the example? Argemone and mustard, that was the question for also for the entrance exam. And we can see that free central. What about free central? That is ovule at the center, and there is no septa, right? Free central example, dianthus primrose. Very important question. And we can see that basal is the next. What about basal placentation? Oval at the base. For the example, sunflower, marigold. And we can see that one more placentation is there, marginal placentation, oval at the ventral suture of the ovary, and marginal placentation is the characteristic features of Fabaceae. We know that. Among these, what's the answer for this question? We can see second, parietal is the correct answer for this question. And what about the next question? Sweet potato is a modified. Sweet potato. Potato is not potato, it is sweet potato. What about that? We can see that. It's a stem. Is it stem? Potato is stem. Potato is underground stem, but sweet potato is not stem. And we know that this is stem. They have nodes and internodes. We know that. Okay, and stem modifications are there, underground, subaerial, aerial stem modifications. An example is very important. And sentence that is given in the NCERT is also important. And next we can see that adventitious root. What about adventitious root? Root form other than from radical. There are modifications also there. We can see that that is tuberculated. That is sweet potato. Yes, that is correct. And next we can see that prop root is adventitious root and still root is their adventitious root. And what about this fasciculated and velamen roots are also adventitious root and hostia is also adventitious root. Yes. And next, that is answer. We know that sweet potato is the adventitious root. That's the answer. Next option. What's the next option? We can see the taproot. Taproot modification we can see that is conical, carrot, CC. Yes, fusiform, radish, FR. Napiform, beetroot, turnip. Very important question for the entrance exam. We can see this term is not in the NCERT, but that will be asked for the entrance exam. Certain chapter is like that. Just give that type of weightage for that chapter. Preparation level you have to be changed while take one each and every chapter. Okay, what are the different questions asked from the previous year? Just go through that, then you can understand. Oh, this pattern will be asked, and what the level I want to be maintained for this chapter. Thus, we can improve the developing. That is, improve or develop our skill for biology. That's important. And next, we can see that fourth option is rhizome. What about rhizome? We know that they are underground stem. Rhizome, bulb, corm, stem tuber, underground, subaerial, runner, sucker, stall, and offset. Then aerial stem, very important. We can see that what is the answer for this question? Yes, of course, two, adventitious root is the correct answer. Okay, and what about the next question? Tricarpellary, syncarpus, gynoecium is found in the flowers of. Very wonderful question, right? Simple question. And we can see that Liliaceae. Yes, we have only three families and one member Fabraceae. And we can see the Liliaceae. What about Liliaceae? They have bracts, actinomorphic, bisexual, perianth. Perianth they have 3 plus 3 fuse. Androecian 3, they are epitapalus, important epitapalus. And that is androecian 3 plus 3. Gynoecium, we can see that tricarpellary, 3 carpels it has. And hypogynous important. We can tricarpellary, syncarpus, fused carpel, yes, of course. Fabaceae 1 carpel, Solanaceae 2 carpel, and Liliaceae 3 carpels. Very easy to study. Monocarpellary, bicarpellary, tricarpellary. Then we can easily connect all these number of carpels. Okay. And what about that's the answer actually, right? This is floral diagram. Floral diagram also very important. Now, for the study of this family, we can write down the floral diagram. We can write down the floral formula and floral diagram, and you can write all the characters into that. Then you can study easily. Otherwise, you will feel more difficult. Okay, just use it. You can do that. You can see the benefit. Okay, and next, second, Solanaceae. What about Solanaceae? We can see that. Yes, floral formula that is actinomorphic bisexual, calyx 5 fused, corolla 5 fused, androecium 5, stamen 5 free, epipetalis important. And then gynoecium that is carpel 2 fused, syncarpus hypogynous. This is floral diagram. We can see that oblique placenta is there. And also we can see that axial placentation is there. We can see that estivation also in this case. Okay, that's important. There's valvate estivation. Here is also valvate estivation. That is an NCERT. And next we can see that Fabaceae. What about Fabaceae? Okay, Fabaceae. What is the floral formula? Zygomorphic. Only remember is zygomorphic for the exam. You have. And this is bisexual S. Sepal 5 fused S. Corolla 1 plus 2 fused. That is called vexillary estivation. Standard wings and fused keel. And androecium, very important. Diadelphus 9 stamen fused plus 1. And gynoecium carpel 1. Hypogynous. Very important question. And we can see that Here, this vexillary respiration, we can see that. Marginal placentation it has, one carpellate it has. And also study economic importance of this chapter, of this area, and this economic importance, there are certain examples that is connected with this family. The question will be like that, given the floral diagram, or given below, no, given below this floral formula, identify the plant which it has. Maybe that plant from that families. That type of questions are there. Therefore, examples, what is given in that area, you have to be study. Okay. Our next option is, we can see that Poaceae. Poaceae family, we don't have, there is no family in the this morphology, but this Poaceae can connect it with the chapter. Can you say? Yes, living world. There is weeds. We can see that. That connection is there. There is no option that is not in the NCRT. Everything is in the NCRT. But that is not in the same chapter. That here and there, scratch from here and there, study. That is the preparation method you have to be developed. And we can see that wheat is triticum, yeast. And we know that they have scutellum, very important. That is scutellum, monocotyledon, single cotyledon, and they are endospermous. And they have aileron layer, that is endosperm and embryo separated from proteinaceous aileron layer, very important. 3N, ploidy is also important, 3N. And spikelate is the inflorescence, connect all the points, what you have, what you can connect. And next we can see that triticum turgidum, that is connected with second year principles of inheritance. Triticum turgidum, that is, we can see that, yes, that is triticum turgidum, autopolyploidy. Right? And then what about apomixis? That is also sexual reproduction, flowering plants. Apomixis by grass, we can see that. That's the point we can connect. And that is from this question, what's the option? What's the answer for this question? We can see that second is the correct answer. Solanaceae. No, first is the correct, that is Liliaceae. Liliaceae, that is tricarpellary, syncarpus, gynoecium. Very important. Okay. Next one, we can see that. What's the next question? The coconut water from tender coconut represents. What is the coconut? Already we know that this is a drupe. Mango and coconut are drupe. In this case, they are this monocot, they are endospermous. This coconut milk, coconut water is liquid, that is nuclear endosperm. And we can see that that is a white kernel of coconut is cellular endosperm, very important. And they have this fruit ball, that is epicarp, mesocarp, endocarp. That is epicarp, we can see that outer coat, epicarp. Then coir making part of the coconut is mesocarp, very important. And inner part is, that is very thick. Endocarp is thick, therefore it is called stony fruits. We can see that. And what is the option? We can see that endocarp. What is the coconut water? We can see that that is endosperm, nuclear endosperm. That is the answer. Is not. Is it endocarp? No. And what about fleshy mesocarp? No. Fleshy mesocarp, that is edible fleshy mesocarp, can be seen in mango. That is also drupe. And we can see that free nuclear proembryo? No. And what about the next? Free nuclear endosperm? Yes. That is the correct answer for this question. And what about the answer? Yes, 4 is the correct answer. And we can see that mango also we can connect with coconut. Drupe. Here mango also we can see that there is endosperm. That is not endosperm, they are dicot. We know the endosperm is consumed, and there is mesocarp is fleshy, and this is epicarp is there, and mesocarp and edible, and endocarp is, we know that is thick, stony fruit. Okay, that's related to these points, this question. And what about, that's about this chapter, morphology of flowering plants. Okay, and next we can move to the next chapter, anatomy. Anatomy of flowering plants. Okay, and what are the different questions? What is the pattern? What is the strength of the question asked? We can see that. In 2019, there are, and 2016, there are 3 questions, and whereas 2018 and 17, there is 4 questions, and 15, there are also 4 questions asked. Okay, very important chapter, we know that. And what about the different concept in this chapter? We can see that there are meristematic tissue, they are primary and secondary meristem. Very important. And next we can see that they are of permanent tissue, they are simple and complex permanent tissue are. And also the tissue system is there, epidermal tissue system is there, ground tissue system is there, and vascular tissue system is there. And also the dicot root, then we can see the anatomy of monocot root, then dicot stem and monocot stem, then dicot leaf is their anatomy. And also we can see that monocot leaf is also there, and also secondary growth. In order to study anatomy, just go through the diagram. Based on the diagram and give all the points in the diagram, then read and study all the points, then you can easily memorize. Based on the diagram, you can easily memorize this anatomy of this dicot root, monocot root, dicot stem and monocot stem, leaf and so on. Okay? And we can see which are the different questions in this chapter. Is 15 the question, Caspian strips occur in. Caspian strips can be seen in. Is Caspian strips occur in? We can see that. Epidermis, is it epidermis? Caspian strips epidermis? Caspian strips is epidermis? Of course not. We know that. And what about pericycle? Is it? No, not in the pericycle. We can see that cortex. No, what is Caspian strips? That is suberinized Caspian strips. It's made up of suberin. Very important. And this what about endodermis? We can see. What about endodermis? Yes. In dicot root endodermis, this Caspian strips present. We can see that. This is Caspian strips. That is suberinized. Part of the endodermis where Caspian strips absent is called passage cell, which you transport from cortex to pericycle. That type of question also there for the entrance. Okay, and we can see that dicot root and monocot. Let's compare dicot root and monocot root. See, from this diagram, we can easily study and what is the features of this anatomy, a dicot root and monocot root. We can see that what is the main difference. The xylem number is only 4 here, but here is more than 6. That is poliar condition can be seen in monocot root. And here is pith is small in dicot root, but here pith is large in monocot root. And here we can see that dicot root there is conjunctive tissue, very important. Monocot root there is no. Okay. And this parenchymatous is hypodermis is there, okay, cortex is there. Okay, fine. Root hair is there. There is no cuticle in the root hair. This epidermis have no cuticle in the root. There is no stomata in the root. That's also important. And next we can see what is the answer for this question is 4 is the correct answer for this question. We can see that. And what about the next question? Question number 16. Secondary xylem and phloem in dicot stems are produced by apical meristem? Apical meristem? No, what about apical meristem? This is apical meristem, that is shoot, salmon, ram, shoot, apical meristem and root apical meristem, and also intercalary meristem also there, meristematic tissue. We know that. And vascular cambium. What about vascular cambium? We can see that. That is cambium that present between primary xylem and primary phloem is vascular cambium, that is intravascular cambium. And there is, we can see that medullary rays in between vascular cambium that is adjoining with intravascular cambium to form intervascular cambium and then become complete ring, that is called cambium ring, that helps in that divide. That helps in the formation of, that leads to secondary growth and helps in the formation of secondary phloem towards out and more secondary xylem towards in, and the secondary xylem is technically called wood. What is the question? Secondary xylem and phloem related to, that is vascular cambium. Of course, the answer is vascular cambium. And what about the next option? What about phellogen? What is phellogen? Yes, that is also helps in secondary growth. We know that is phellogen is cork cambium. Towards outer that is produce phellum or cork, and towards inner produce phellodame or secondary cortex, and three together called phellum, phellogen, phellodame called peridame. Peridame plus secondary phloem is called bark of the tree. That's very important question for the entrance. And next option, what is the next option? Axillary meristem. What is the point related to axillary meristem? We can see that left behind from shoot apical meristem become axillary bud. Very important point. And this axillary bud is helps in formation of branch or flower. That's also a question for the entrance exam. And we can see that what's the answer for this question? Second option is the correct answer for this question. Okay, and what about the next question? Grass leaves curl inwards during very dry weather. Select the most appropriate reason from the following. And what we can see, first option, flaccidity of bulliform cells. What is bulliform cells? We can see that they are large, empty, colorless cells that is formed in the adaxial surface, that is upper epidermis of leaf in grass. And that is about, what is the purpose? Control the process of transpiration. We can see that. That is, of course, that is, yes, this is bulliform cells. We can see that. When we become flaccid, that curl leaf inwards and reduce transpiration by the grass. That is, it can be seen in grass, very important. That is where present, that is also asked for the question, that is also entrance question, adaxial. Abaxial is base, lower. Okay, and next we can see that what is the option? Second, shrinkage of spaces in spongy mesophyll. What is this? What is mesophyll? Leaf cortex. What is cortex? Leaf cortex, that is upper epidermis, lower epidermis, between that. And this, there is palisade and spongy parenchyma in the mesophyll of dicot. Whereas in monocot leaf, there is no such distinction. We can see that. And next can see that. Tylosis in vessel. What is tylosis? We can see that. That is, tylosis is the outgrowth of xylem parenchyma into the vessel in the heartwood in order to injury, that is response to injury or protection from decay in heartwood. We can see this is tylosis, xylem parenchyma into the vessel. That is called tylosis, balloon-like structure. Yes, that's important question for the entrance. And next option, option 4. What about this? Closure of stomata. What about stomata? We can see that this is a dicot stomata, kidney or bean shape, whereas monocot, dumbbell-shaped stomata. We can see that this, how it is, what is the structure? Inner wall is thick and elastic, outer wall is thin. They have cellulose in microfibers, radially arranged cellulose in microfibers, helps in, that is opening, that is maintain this longitudinal opening of the stomata, stoma opening. And also this inner, since this thicker inner, this inner wall is thick and elastic, therefore it is turgidity can be maintained. This turgidity, because of this turgidity, this guard cell become, because of the turgidity of guard cell, stomata will be opened. And when this stomata will become flaccid, that is guard cell become flaccid, what happened? This stomata become closed. We know that. Okay, and the question is also like that, facilitate opening of stomata by this, because of the radially arranged cellulosic microfibers also was there for the answer. Okay, here we can see what's the answer for this question. Yes, 1 is the correct answer for this question. And what about the next chapter? We are going to deal next chapter. What is that? We can see that cell, the unit of life. And while go through this question, we can see previous questions, we can see that in 2019, 18 and 15, there are 4 questions asked, whereas in 2017, 1 question asked from this chapter. This chapter is also very important. We know that actually this chapter deal with these 2 types of cell, prokaryotic and eukaryotic cell. This prokaryote can connect with kingdom Monera. Okay, therefore certain points are also there. We can see that. And we can see that prokaryote have no true nucleus, they have no membrane-bound organelles. And here we can see that there is true nucleus and have membrane-bound organelle. They have endomembrane systems such as endoplasmic reticulum, Golgi apparatus, lysosome, vacuole. We know that. There is chloroplast, then mitochondria. Then we can see that peroxisome, that is microbodies are there. We know that. That is the case of eukaryotes. Every point you have to be studied in the NCERT. Okay, diagram is also very important. Every diagram, you have to go through the every diagram, that's very important. And what are the different questions from this area? The concept of omnicellular cellulae regarding cell division was first proposed by, what are the different points? Yes, Theodor Schwann, that is the option of Theodor Schwann. In the NCERT, we can see the year 1839, and he is a British zoologist. Very important question for the entrance exam. British zoologist, and reported plasma, that is in animal cell have plasma membrane, and the plant cell wall is also reported by this Theodor Schwann. Very important, right? Very important concept. And we can see that hypothesis that bodies of animals and plants are composed of cell and product of cell by Theodor Schwann. He is a British zoologist in 1839. And next option, we can see that Schleiden. What is the full name of Schleiden? Matthew Schleiden in 1838. And what's the contribution? We can see that he's a German botanist. He's a German botanist. And we can see that all plants are composed of different kinds of cell which form the tissue of the plant. Very important question for the entrance, or the statement question will be asked from this area. And we can see that Schleiden and Schwann together formulated cell theory. That question also there. Who formulated cell theory? Schleiden and Schwann. And what's the next option? Aristotle. What about Aristotle? We know that he is father of biology and zoology, and we know that. And he, or one point, there are another points that we can see that animal with red blood and without red blood by this Aristotle, and also plants classified into trees, shrubs, and herbs. So by Aristotle, these are all other points in the NCERT. Very important point. And what about the 4th option? Rudolf Virchow. Rudolf Virchow, very important. We can see that is in 1855 explained that cell is divided and new cells are formed from pre-existing cell. That is called omnicellular cellulae. Omnicellular cellulae, that is 1855. Rudolf Virchow, very important. Very important. And next we can see that he modified the hypothesis of Schleiden and Schwann to give cell theory a final shape. Who modified cell theory? Rudolf Virchow. And who formulated cell theory? That is Schleiden and Schwann. What is written in the NCERT? That will be the answer for the Neet exam. There is no doubt. Okay, what is the answer for this question? Omnicellula, ecellulae. Of course, Rudolf Virchow. Answer 4 is the correct. It's the correct answer. And what about the next question? Which of the following statement regarding mitochondria is incorrect? Why I bold this? Not to notice incorrect or correct, true or false. Very important. Avoid careless mistake. Please avoid careless mistake while in the exam hall. Please avoid hurry in the exam hall. Very important. Keep calm and prepare and write down every point. No, don't worry. You can win. You can win. There is no doubt. Okay, and we can see that. And what is the structure? We can see what is the structure of mitochondria. We can see that. There is outer and inner membrane. Inner membrane has folding is called cristae. And inner membrane has F0 F1 particle for terminal oxidation. ET, yes, electron transport chain or electron transport system. Yes. And we can see that this inner membrane there is called matrix. Inside matrix, we can see that DNA is there, ribosome is there. This is semi-autonomous organelle. Ribosome is the organelle within an organelle. That can be seen in mitochondria and chloroplast. Okay, we can see that. First option. Enzyme of electron transports are embedded in outer membrane. That is electron transport is ETS. Is it outer membrane? No, in the inner membrane. That is wrong. What is the question? Incorrect. Which is incorrect? First option itself is incorrect. We know that. And what about second? Inner membrane is convoluted with infolding. Yes, of course. That is called cristae. But cisternae can be seen in Golgi apparatus. But it is cristae. Okay, read the word and term carefully. This is very important. Next, we can see that mitochondrial matrix contains single circular DNA, yes. Molecule and ribosome, yes. Single circular DNA molecule and ribosome, yes. Correct. And next, we can see that outer membrane is permeable to monomer of carbohydrate, fat, and protein, yes, of course. What's the correct answer? What is incorrect? Which one is incorrect? One is incorrect for the question number 19. And what about the next question? We can see that question number 20. Which of the following statement is not correct? It's not correct. In the previous question, incorrect. It's not correct like that. The hydrolytic enzymes of lysosome are active under acidic pH? Of course, correct. And next, this is, we can see that from the endoplasmic reticulum, protein will be synthesized and transported to the Golgi apparatus. There is formation and maturation packing will be there in the Golgi apparatus. From their release product is called lysosome. We know that they are membrane bound structure. Lysosome, zooidal bag, lysosome, they are hydrolytic enzyme there. Okay, they are okay, we can see that. And what about the second option? Lysosomes are membrane bound structure, of course. They're membrane bound structure. And next, lysosomes are formed by the process of packing in the endoplasmic reticulum. Is it packing in the endoplasmic reticulum? Of course not. What is the correct? That is what is which where it is packed? In Golgi apparatus. Therefore, that is not correct. Third option is not correct. Question is not correct. Which is correct answer? 3 is the correct answer. And what about the question number 4? Lysosome have numerous hydrolytic enzyme, of course. Protease, lipase, nuclease, etc. there in the lysosome. We know that. And what's the answer for this question? We can see that this 3 is the correct answer for this question. Okay. And we are providing now long term, that is +2 courses also there, and everybody have to be prepare all these points, all these entrance points thoroughly, and what are the different points we discuss, and read NCERT thoroughly, and that's very important. That's only the method you get full marks for biology. Okay, and next we can see that. This next question, 21, question number 21. The shorter and longer arm of metacentric chromosomes are referred to as. Yes, very important question. We can see that. This is chromosome morphology. Chromosome can be distinguished on the basis of size and the relative location of centromere. The centromere is here. Based on the location, we can. There are, so that is, which are the different type of chromosome there is. We know that metacentric, submetacentric, acrocentric, and telocentric. In the submetacentric chromosome, we can see that the question is submetacentric. We can see that there is short arm, there is petit. It's a French term, short arm, P. Therefore, we can give Q for long arm, P and Q arm. We can see that in submetacentric and acrocentric. We can see that submetacentric and acrocentric. We can see that. Okay, and what is the options? P arm and Q arm respectively? Yes, that's the correct answer. This is NCERT based preparation. Okay, P and Q terms are not in the NCERT, but if you are preparing based on NCERT, you can find out that term also. There is no doubt. And next we can see that Q arm and P arm respectively? No. Longer arm is longer, shorter is P and longer. That is the option. Shorter and longer respectively if there. That is referred to as that is respectively in the answer. If correct order have to be there in the answer. That is P and Q arm. And what about the third one? M arm and N arm? No. And what about the fourth one? S arm and L arm? No. What is the correct answer? S1 is the correct answer. We can see that. And what about the next question? That's next chapter. We are going to deal next chapter. That is biomolecules. So it's a very simple and important chapter. We can see that. While go through the previous question, we can see that. In 2018 and 15, there are one question, and 2004, that is 19, 4 questions, and 2016, there are 3 questions from this chapter. Important chapter and simple chapter. Easy chapter, we know that. And this primary and secondary metabolites are there. Study all the points and all the tables in the chapter, and amino acids are there, and we can see that lipids are there, and nucleotides are there, and we can see that there are proteins, polypeptides are there, and we can see that polysaccharides are there and monosaccharides are there, and we can see that nucleic acids are there in this chapter, and also enzymes are also there. Okay, which are the different questions in this chapter we are going to see? With reference to enzymes, which one of the following statement is true? Yes. Now we can see that this is apoenzyme and this is protein part of the enzyme is apoenzyme. And this non-protein part of the enzyme is called cofactor. Cofactor of 3 types. We know that prosthetic group, coenzyme, and metal ion. Prosthetic group are organic compound tightly bound with protein part. Coenzymes are protein, that is organic compound loosely packed with, loosely associated with the protein part, that is apoenzyme. While they are become apoenzyme and cofactor join to form holoenzyme, whole enzyme that is active. Based on this question is there. What's the question? We can see that apoenzyme is equal to holoenzyme plus coenzyme. Apoenzyme is equal to holoenzyme plus coenzyme. No, that is not correct. Second, holoenzyme is equal to apoenzyme plus coenzyme. Coenzyme is cofactor. Yes, correct. Apoenzyme plus coenzyme, that is cofactor. Correct. That's correct. This is the correct one. If you understand this concept, then you can answer correctly. There is no doubt. And next, that is the next option. Coenzyme is equal to apoenzyme plus holoenzyme. No. Coenzyme is equal to apoenzyme plus holoenzyme. No, that's not correct. And what about fourth one? Holoenzyme is equal to coenzyme minus apoenzyme. No. What's the correct answer? Second is the correct answer for this question. And what about the next question? Which of the following are not polymeric? Polymeric, monomer, group to form polymer. First option, nucleic acid. What is nucleic acid? They are polynucleotides, DNA and RNA. Nucleotides are interconnected by phosphodiester bond. Nitrogen base plus sugar by N-glycosidic bond to form nucleoside. Nucleosides are interconnected to, that is associated with phosphate by phosphoester bond to form nucleotides. We know that. Very important bonds in the biomolecules. And next we can see that proteins. What about proteins? There are primary, secondary, tertiaries, quaternary structure of protein, and the proteins, building blocks, and amino acids interconnected by peptide bond. They are polypeptide. Proteins are polypeptides. We know that. And polysaccharide. What is polysaccharide? Monosaccharide associate become polysaccharide. We know that. The bond is glycosidic bond. Monosaccharide interconnected by glycosidic bond. We know that starch is there, cellulose is there, then glycogen is there, inulin is there, Chitin is there, and the NCRT, very important. Study all the points. They are homopolymer, but protein is heteropolymer. But these are homopolymer, polysaccharide. All these are polymer. Next option, lipids. What is the specialty of lipids? They are not. They are not become polymer. They are not become polymer lipid. We know that. They are simple lipid and compound lipid. And we know that we will see in the next slide. And we can see that what's the answer for this question? Lipid is the correct answer for this question. Is 4 is the correct answer. And we can see that lipids, simple lipid, compound lipid, and we can see that there is derived lipids are there, cholesterol is there, compound lipids, phospholipid, very important, lecithin, phospholipid, very important question for the entrance. Yes, and we can see that this is saturated and unsaturated is also there. Fatty acid, very important, very important. Okay. Next question. The two functional groups characteristics of sugars are, this is sugar structure. We can see that there are two functional group, carbonyl or aldehyde and hydroxyl group are there. Yes, that is a group in the sugar. That is the option. What's the option? Option 1. Carbonyl and phosphate. No. Next. Carbonyl and methyl. No. Next. Hydroxyl and methyl. No. What's fourth option? Carbonyl and hydroxyl. Yes, that's the correct answer. We can see that sugar is common term used to denote carbohydrate. Carbohydrates are polyhydroxyaldehyde or ketone or their derivatives, which means they have carbonyl and hydroxyl groups. Very important. And what's the answer for this question? 4 is the correct answer for this question. And then we can see that. What's the next question? Which one is the abundant protein in the animal world? We know that. What's the answer? Trypsin. Trypsin is the enzyme. We know that. Next, hemoglobin. We know that hemoglobin is the pigment, is a protein. Hemoglobin, adult hemoglobin consists 2 alpha and 2 beta subunits, quaternary structure. This alpha thalassemia, beta globin chain affected sickle cell anemia. Connect like that. Connect like that. Okay, and next we can see that collagen is the abundant protein in the animal world. And next is, which is the abundant protein in the biosphere? Rubisco, ribulose, bisphosphate, carboxylase, oxygenase, activated by magnesium. Yes. And insulin. Insulin is the hormone that is connected with biotechnology, insulin, humulin, right? And we can see that what's the answer? 3 is the correct. Collagen is the abundant protein in the animal world. And we can see that this type of tables are in the NCERT. Study all the tables. Maybe that will be asked for the match the column or mismatch pair, one word questions where from these chapters. Study all these points. Okay, everything. Glut for enable glucose transport in the cell, and what about the percentage of carbohydrate in the cell? 3. These type of questions are there. Okay, we can move to the next chapter. What's the next chapter? Yes, cell cycle. While go through the previous questions of cell cycle, we can see that yes, in 2019, 3 questions, and 2018, 1 question, and in 16, there is 4 questions asked, and 15, there is 2 questions asked from this area, from this chapter, very important chapter. There's a certain, very simple chapter, and there is certain diagram is there, so concept is there, study all these things. And we can see that. Which are the different concepts in this chapter? Interphase, G1S, G2. Yes, and M phase, division phase, mitosis and meiosis, consists karyokinesis and cytokinesis. We know that. This is mitosis, consists prophase, metaphase, anaphase, telophase. The segregation of chromatids takes place during anaphase. After mitosis, dyad of cell, 2 daughter cells will be formed. We know that second, meiosis. We can see that meiosis. Meiosis 1 and meiosis 2 is there. Phase between meiosis 1 and meiosis 2 is called interkinesis. And after meiosis, there are 4 daughter cell, tetrad of cell with haploid chromosome number will be there. Diploid organism meiosis will be there. Okay, we can see these concepts while discussing the questions. Question: the stage during which separation of the paired homologous chromosome begins. Separation of paired chromosome begins, that is connected with meiosis 1, prophase 1. Yes, we can see that. What's the first option? Packeting. What's the bond-related to packeting? Packeting, recombination takes place during packeting. Recombination takes place during packeting. And tetrad can be clearly visible during packeting. Very important question for the entrance. Tetrad can be clearly visible during packeting. And next, Diplotin. What is diplotin? We know that dissolution of synaptonymal. That is synaptonymal complex will be dissolution that form chiasmata. X-shaped structure that is called chiasmata. That is separation of paired homologous chromosome begin during diplotin. Diplotin begins during diplotin. Yes. Third option, diakinesis. What about diakinesis? That is terminalization of chiasmata. Terminalization of chiasmata during diakinesis. Very important question for the entrance. And also next option, zygote. What happened during zygote? Synapsis. Pairing of non-sister homologous chromosome. Pairing of non-sister homologous chromosome. Bivalent formation takes place during zygote. Very important. And one more stage in this prophase. One is leptotin. We know that. Leptotin, zygote, pachytene, diplotin, diakinesis are the stages of prophase one, of meiosis one. We know that. What are the options? What is the question? Paired homologous chromosome separation begin during diplotin. Second option is the correct answer. And what about the next question? The correct sequence of phases of cell cycle is, we can see that G1. What is G1? No, there's a points. After discussion of these points, we will go through the options. The options. From there, we can understand. G1. What about G1? No, the cell is metabolically active. And if the cell is not to divide, again, it will enter G0, quiescent stage. We can see that. Then what about S, synthetic phase? We can see that. Amount of DNA is double, synthesis of DNA. We can see that. And the number of chromosomes remain constant during S phase. We can see that. And we can see that G2. During G2, proteins are synthesized in preparation for mitosis while cell growth continues. And M phase is there, that is called, we can see that is the cell divides, it's copy DNA and cytoplasm to make two new cells. M phase, that is karyokinesis, and cytokinesis will be takes place. What are the sequential stage? We can see that this one, G1, S, G2 are the sequential stage. And then there is M phase. M phase consists karyokinesis and cytokinesis. Karyokinesis are prophase, metaphase, anaphase, telophase. Okay? And then cytokinesis is cytoplasmic division. This is the cell cycle representation. We know that cyclin-dependent kinase is an enzyme which controller, who is the controller of cell cycle. And what is the sequential stage? G1S, G2 and M. G1S, G2 and M. What about the option? Option 3, G1S, G2 and M is the correct answer for this question. What's the correct answer? 3 is the correct answer for this question. G1S, G2 and M. While go through this concept, while go through this SG1S, then just think about what are the points I have to be memorized. What are the points important for entrance? Which diagram I have to be memorized. These types of preparation you have to be maintained during these days. Okay? And next question. Which of the following options gives the correct sequence of events during mitosis? Mitosis. We know that mitosis consists prophase, then metaphase, anaphase, telophase. You know the process. Metaphase, equator, then anaphase, that is centromere, that is divide, and chromatids move to the two opposite poles during anaphase. Okay, what are the sequential stage? Very important question. We can see that first one, condensation. Condensation happened here. Next, nuclear membrane disassembly. Yes, hide. Yes, that is membrane will become hide. Membrane become there, not there now. Okay, yes, correct. Arrangement equator, that is during metaphase. Yes. Then centromere division, yes, during anaphase. And segregation, yes, segregation, yes. Chromosome, chromatid segregation, and then telophase. Yes, that is the correct order. One is the correct order. What is the correct answer? And this is the correct answer. What's the correct answer? We can see that 1 is the correct answer. Every option have to be read carefully, and concept clarity is very important. Concept clarity is important. Okay, we can see that that's about cell cycle. Now we are going to discuss next chapter, transport in plants. We are going to move plant physiology, transport in plants. From this chapter, but while go through the previous question, we can see that 2016-15 there are one question asked, whereas in 2019-18 two question asked. Yes. Important chapter. We can see that which are the concept, means of transport, simple diffusion, facility diffusion, active transport. And next we can see that Water potentials, potential of water to flow, or kinetic energy, or concentration of water, or free energy of water. We know that. Water potential, the water movement process, osmosis. Osmosis, we know that there are plasmolysis, exosmosis, turgidity, endosmosis, and imbibition. Yes, next we can see that mass flow for long distance of transport. We can see that, and absorption and transport of water. Absorption, very important. Apoplast, symplast pathway, very important. And we can see the ascent of sap, guttation, root pressure theory, and transpiration pull theory, very important. Dixon and Joly, Dixon's theory of ascent of sap, important point. And we can see that phloem transport, that is soul sink, very important. And we can see that phloem transport girdling experiment, and we can see that Munch hypothesis, mass flow or pressure flow hypothesis, that is phloem transport. While discuss the question, we can see certain points. Okay, and what are the question? Xylem translocates. Xylem translocates. Option 1, water and minerals, of course, but in the NCRTs, one sentence is there. Based on that, this question is asked. And what's the second option? Water and mineral, salt, and some organic nitrogen only? No. Third, water, mineral, salt, some organic nitrogen, and hormone. While read the NCRT, you can see the same sentence. Same sentence in the NCRT will be asked for the Neet exam. There is no doubt. Okay. And next, water only? No. What's the answer for this question? Third, xylem unidirectional. We know that. Yes. What's the answer? 3 is the correct answer for this question. And what about the next question? Question number 30. The water potential of pure water is, water potential of pure water is less than 0? No, less than 0 can be in solute potential, negative. And more than 0 but less than 1? No, no, that's not correct. More than 1? No. Usually positive, that is water potential, that is pressure potential is usually positive. Pressure potential is usually positive. And 0, yes, of course. Water potential of pure water, STP is 0. Very important. Water potential, that is psi W is equal to psi S plus psi P. Psi S is always negative. Psi P is usually positive. Very important point. Very important point. Okay. Next, what's the answer for this question? 0 is the correct answer. 4 is the correct answer. And next one, we can see that. No, we can see that. No, the next, before going to next question, we can see that's long term. We have also conducted this +1 courses also there. Okay, and we are providing all opportunity. Use these opportunities. Okay, and use all the opportunities. And next we can see that a few drops of sap were collected by cutting across a plant stem by a suitable method. The sap was tested chemically. Which one of the following test results indicate that this phloem is a phloem? While reading question, no, I get confusion, no? But it is simple concept. What's the question? Phloem sap means which one is transported to phloem? That is through sieve tube. And that is sucrose. In which form food is transported through sieve tube? That is sucrose. What's the nature of sucrose? That's the question. Okay, and we know that the sucrose transported through sieve tube by mass flow or pressure flow. That is discovered by Munch, therefore it is called Munch hypothesis. We know that. And what's the point? Low refractive index? No. Second, absence of sugar? No. Acidic? No. Third, alkaline? Yes, that's the correct answer. Why? Phloem sap is generally alkaline, which pH ranging from 7.3 to 8.5, and contains high level of potassium and magnesium also. That is why it is alkaline. Okay, therefore answer is 4. And what about the next question? Next chapter we are going to deal, next chapter, mineral nutrition. While go through the previous question, we can see that 2001, that the 1918 one question asked, and whereas 2016-15 2 questions asked. Very important chapter. Simple chapter, but important chapter. Certain functions of minerals are there. Every point, microscopic study is important. Microscopic study is important. Okay. And what about the question? We can see that. Which are the different points? Hydroponics is there. Julius von Sachsen, 1860, very important. German botanist. And we can see that next is macro and micronutrients. More than 10 mol per kilogram of dry weight is macro, less than 10 mol per kilogram of dry weight is micronutrients or trace element, very important. And we can see that role, very important role, very important. Study all the points in the NCRT. And next we can see that, yes, deficiency symptom, We can see that delay flowering, chlorosis, then that is, we know that cell division inhibition, necrosis, premature leaf fall. These are the different points in the NCRT. And toxicity, manganese toxicity leads to the deficiency of magnesium, iron, and calcium. Symptom also there. Right. Next, we can see that absorption and transport. Absorption by active and passive. And there is apoplast, symplast. Apoplast is free space, and symplast is inner space. Yes, very important. And we can see that nitrogen metabolism, there is, we can see that nitrogen fixation, and then nitrification, then ammonification, and denitrification are the process of nitrogen fixation. We can see that. There are the different points in that chapter. So the different question we are going to see. In which of the following form iron is absorbed by the plant? These type of questions are also there. Okay? And what's the point? Ferric. Ferric, iron absorbed in the form of ferric. Yes, Fe3 plus ferric. Yes, correct answer. Ferric, correct. Is it ferrous? No. Is it free element? No. Is it both and ferrous and ferric? Which is the correct answer? We can see that. Yes, one is the correct answer. Different absorbable form you have to be studied. Nitrogen absorbed in the form of nitrate. Phosphorus H2PO4 minus or HPO4 2 minus. Boron BO4 2 minus. Very important. BO4 2 minus. And we can see that SO4 2 minus is, we can see that this sulfur. And H2PO4 minus, phosphoric acid, very important. Nitrogen, nitrate is the main absorbed form, cannot absorb as ammonia, very important. Can absorb as ammonium also, but ammonia is not there. Rarest form, ammonia. Ammonium can never absorbable form, ammonia. Therefore, after nitrogen fixation, ammonium is converted to nitrate. That is the most absorbable form. That process is called nitrification. We know that. And we have to be study all the points, all the absorbable form thoroughly, very important. Okay, and we can see that. What's the question? Which of the following element is responsible for maintaining temperature in cell? Magnesium. What about magnesium? We can see that. So what is the magnesium? What is the role of magnesium? Very important. Photosynthesis, active enzyme for photosynthesis, respiration, synthesis of DNA, RNA, magnesium. And magnesium maintain ribosome structure, porphyrin head, pyrrole ring structure maintained by magnesium. And also this magnesium helps in ribosome structure, very important. And helps in synthesis of chlorophyll. That's the question for the entrance. And sodium is beneficial element. Which are the beneficial element? Cobalt, sodium, silicon. Is very important. And next we can see that potassium. Potassium maintain anion cation balance, stability of gut cell. That's the answer for this question. And we can see that potassium that is maintained this protein synthesis also. You can read NCRT. We can see that potassium activate enzyme. That's important. Potassium very important point. All the function very important. Thoroughly study. And we can see that calcium. Calcium cell division. Calcium. Then mitotic spindle formation. Calcium pectate. Then we can see that regulate metabolic activities. Uptake and utilization of calcium by boron. What about boron? That is pollen germination, cell elongation, cell differentiation, membrane functioning, carbohydrate translocation by boron. And these type of memory tips you have to be developed. And nitrogen, we know that very important. PNVH, protein, nucleic acid, vitamin, hormone by nitrogen. Phosphorus, phosphorylation. And we can see that molybdenum and nitrogen metabolism, very important. And iron helps in iron catalase, alcohol dehydrogenase, zinc, molybdenum, nitrogenase, important. Okay, and we can see that synthesis of chlorophyll also by iron. Main is magnesium. Okay, and next, these are the functions. Thoroughly study every point. Very, very important area, that area. And what's the answer for this question? 3 is the correct answer for this question. And next we can see that. Select mismatch. Select mismatch. And what about rhodospirillum? Mycorrhiza. What is rhodospirillum? We can say rhodospirillum. What is the point? Is anaerobic free living rhodospirillum and clostridium. Anaerobic free living rhodospirillum and clostridium. That is not mycorrhiza. Mycorrhiza, what is? That is fungi come under glomus genus associated with the pinus root to form mycorrhizae. For mineral, that is phosphorus absorption, that is mycorrhizae. That is connected with worm, vesicular, arbuscular mycorrhizae. Okay, that is endomycorrhizae. Ectomycorrhizae is also there. That is not correct. And next we can see that anabina. Anabina, nitrogen fixer, is correct. Which is mismatch? Of course, rhodospirillum is mismatch. But anabina, nitrogen fixer, correct. Anabina azola. Anabina is cyanobacteria. Cyanobacteria, photosynthetic atrophy bacteria. Anabina with azola, no stroke with anthoxyrose, symbiotic nitrogen fixer. Very important. Next we can see that rhizobium. Alfalfa, rhizobium is, we can see that is That is present in leguminous plant, alfalfa, lentil, sweet pea, sweet clover, garden pea, beans, all these are leguminous. That is enter into that plant, fixed nitrogen. Correct. Rhizobium alfalfa, correct. And what about Frankia? Frankia is filamentous bacteria that is enter into the non-leguminous plants such as Ulmus and Casuarina helps in symbiotic nitrogen fixation. Very important question. Very important point. What's the correct answer? One is the correct answer. Which one is incorrect? One is incorrect. And next we can see that. Next chapter. Photosynthesis in higher plants. That's also very, very important chapter. We can see that while go through the previous question. In 2016, there are 4 questions, and 2017, 1 question, and 2019, 18, there is 2 questions asked. Very important. We can see that. These are the different concepts in this chapter. Experiments, thoroughly go through the experiments. Year, very important. And experimental material used also important. And light reaction, we know that cyclic and non-cyclic photophosphorylation are there. Study the point, and we can see that dark reaction in order to synthesize glucose by using the assimilatory power which is produced by light reaction, that is C3, C4 and CAM pathway, that for the production, that is for the production of glucose, we know that. And photorespiration, that is C2 pathway, that can be shown, that is can be present in C3 plants. And next we can see that these are the different factors affecting photosynthesis, and also law of limiting factor by F.F. Blackman in 1905. Important point, we can see that. Which are the different questions? Which of the following is not a product of light reaction of photosynthesis? Light reaction, very important. Light reaction, cyclic. What is cyclic? Yes, we can see that. This PS1 only there during cyclic reaction, and light absorbed by PS1 and expelled electron from this PS1 and moved to the electron transport system and returned back to the same chlorophyll, that is PS1. Therefore, it is called cyclic, but phosphorylation, that is phosphorus is added to ADP to form ATP, that is phosphorylation takes place. That is called cyclic phosphorylation. That happened in stroma lamella or Fret membrane. Yes, that is called cyclic photophosphorylation. Only ATP is produced. And what about non-cyclic? Here PS2 and PS1 is there. Both pigment system accepted this light, and electron expelled from this photosystem and accepted by electron, accepted and transported through electron transport system. And we can see that phosphorylation to ADP and to form ATP. ATP will be formed, and from this PS2 electron expelled, electron accepted by NADP. NADP is reduced to NADPH plus H plus. And we can see that from PS1 to PS2 to PS1, from PS1 to NADPH, from water to PS1, electron is moved. Therefore, electron is not returned back. From expelled electron from the PS1 is not returned back to itself, that is moved to the NADP. From PS2 to PS1, from PS1 to NADP, therefore it is called non-cyclic. But here it is ATP formation is there, that is called non-cyclic photophosphorylation in the presence of light. That is explained by how it is photophosphorylation takes place, explained by chemiosmotic hypothesis by Peter Mitchell. Important point. We can see that during non-cyclic photophosphorylation, we can see that there is ATP will be there, NADPH will be there, and water splitting photolysis of water with help of element, chlorine, magnesium, chlorine, manganese, calcium, very important. Chlorine, manganese, calcium are the element helps in photolysis of water, very important question. And activate enzyme for photosynthesis, respiration by magnesium and manganese, that type of question also there. And we can see that here oxygen is evolved. Photolysis of water that takes place during non-cyclic. And what is the product of light reaction of photosynthesis? Which is the following is not a product? That's the question. Among these, which one is not can see? We can see ATP, yes, of course, ATP is there. And second option, NADH, no. What is NADH? Nicotinamide adenine dinucleotide. That is the reducing enzyme participate in catabolic process. We know that photosynthesis is anabolic process. In this catabolic process, we know that is respiration. The respiration, we can see that NADH is there. Here there is no NADH, therefore answer is NADH, of course. NADPH is there. What is NADPH? Nicotinamide adenine dinucleotide phosphate. That is also reducing coenzyme participate in anabolic process. And what about the fourth one? Oxygen. Yes, oxygen is produced. Which is the answer? Second is the correct answer. Second is the correct answer for this question. And what about the next question? Cyclic photophosphorylation result in the formation of, we had seen that. Cyclic phosphorylation, NADPH, no. NADPH only during non-cyclic. And ATP and NADPH? No, that is during non-cycling. And ATP, NADPH and O2? No. And ATP, yes. What's the correct answer for this question? Yes, ATP. ATP, only ATP is produced during cyclic photophosphorylation. Answer is 4. We can see that 4 is the correct answer for this question. And what about the next question? Phosphoenol pyruvate, that is PEP, is the primary carbon dioxide acceptor in. We can see that. Option 1, C4 plant. What about C4 plant? We know that they have crans anatomy. Mesophil, that is bundle sheath is surrounded by mesophil like ring or wreath or crans. Therefore, it is called crans anatomy. We know that. They have two cells in order to complete their photosynthesis. They have mesophil and bundle sheath cell. This mesophil, we know that there is pepcase, phosphoenol pyruvate carboxylase, accepted carbon dioxide that is entered into the carbon dioxide entered into the cytoplasm and dissolved in water to form bicarbonate, accepted by PEP and to form fixed to form oxaloacetic acid, first stable product. 4-carbon compound that is reduced to form malic acid that is transported to bundle sheath cell and where it is decarboxylated, release carbon dioxide accepted by Rubisco enter into the Calvin cycle. Calvin cycle takes place in bundle sheath cell. And we can see that require for this Calvin cycle 3 ATP and 2 NADPH. And this pyruvate, 3-carbon pyruvate is back to the mesophil and regenerated by using 2 ATP and produce PP. That is one cycle. Thus, 6 cycles stand, 1 glucose will be formed. For 1 glucose formation, there are 30 ATP and 12 NADPH required. We know that. That's about C4 plants. Phosphoenol pyruvate is the primary carbon dioxide acceptor in C4 plants. We can see that. Okay, and next option. C2 plants, there are no such plants. C2 pathways are there, that is called photorespiration, that takes place in C3 plants. When oxygen condition, oxygen amount is more than carbon dioxide. That is, we can see that which are the organelle participate, that is mitochondrion, peroxisome and chloroplast, the organelle participate in this process. Very important, 3 organelle participate during photorespiration and this reduces the efficiency of C3 plants. But this photorespiration absent in C4 plant. Very important point. And next we can see that C3 and C4 plants. No, not in C3 plants. And what about next? C3 plants. No, there is no C3 plants. We can see that this is, we can see the mesophil cell only is there for photosynthesis. There is chloroplast, mesophil chloroplast is there. There is no crans anatomy. Then we can see the Rubisco is there, which accepted carbon dioxide, then carboxylation, RBP, and to form 3-PJ, 3-phosphoglyceric acid, 3-carbon compound. And we can see that that is undergo reduction by using ATP and NADPH and form triose phosphate and again regenerated REBP by using 1 ATP. Per cycle we can see 3 ATP and 2 NADPH used. 6 cycles are used to complete not to form 1 glucose. Therefore 18 ATP and 12 NADPH required and we can see that after formation of glucose, glucose is transported in the form of sucrose and sucrose that is through sieve tube and stored in the form of starch. The case of plants. Okay, therefore, we know that we have, that is CAM plants also, that is crassulation as in metabolic plants, that is pathway is also there. And what's the answer for this question? Is 1 is the correct answer for this question. Next, we can see that, question number 38. With reference to factors affecting the rate of photosynthesis, which of the following statement is not correct? Yes, we can see that. Light saturation for carbon dioxide fixation occurs at 10% of full sunlight. Is correct. That is also connected with light, area light. Okay, and this is very important. Is correct sentence. Increasing atmospheric carbon dioxide concentration up to 0.05% can enhance carbon dioxide fixation rate. Is correct. Is correct. Next. C3 plants respond to higher temperature with enhanced photosynthesis, while C4 plants have much lower temperature optimum. No, what's the answer? We can see that C3 plants that increase at higher temperature, a rate of photosynthesis in C3 plants increase at higher temperature due to increased photorespiration. And we can see that decrease in the case of C3 plants. But in the case of C4 plants, higher temperature optimum because of the presence of this enzyme, pyruvate kinase enzyme, and which is sensitive to low temperature, PP. That's the point. Therefore, this is wrong. This is wrong. And what about the fourth statement? Tomato is a greenhouse crop which can be grown in carbon dioxide enriched atmosphere for higher yield. Tomato and bell pepper. Greenhouse crops. Very important. We can see that from this. Yes. 3 is the correct answer. Internal, intrinsic, and external. Extrinsic factors are there. Internal, that is, we can see the number, size, orientation of leaves, canopy structure, internal carbon dioxide concentration, number, chlorophyll, chloroplast, etc. And external factors, light, temperature, water, carbon dioxide. Very important points. Okay. Next, we can see that that's about this chapter. Next, we can move to the next chapter. What's the chapter? Yes, respiration in plants. And while go through the previous questions, we can see that in 2018, 17, there are two questions asked. Very important chapter, respiration in plants. We can see that. Which are the different concepts in respiration in plants? Anaerobic respiration. Anaerobic respiration are of alcoholic fermentation and lactic acid fermentation. During alcohol fermentation, we can see that alcohol will be produced anaerobic respiration. And first process of anaerobic respiration is also glycolysis. And we can see the aerobic respiration. Which are the sequential steps of aerobic respiration? What is the specialty of aerobic respiration? Complete breakdown of food takes place during aerobic respiration. We can see that the presence of oxygen is there. And we can see that. Show the process. First one is glycolysis. Glycolysis. What is glycolysis? We know that glucose is converted to pyruvic acid. That is what is the net product of glycolysis? 8 ATP is the net result of glycolysis. Very important point. Yes. And what about link reaction? That is called, that is oxidative decarboxylation that happen in mitochondrial matrix. That is net result is 6 ATP. Yes, that is pyruvic acid is converted to acetyl coenzyme A. And we can see that Krebs cycle. Acetyl coenzyme is completely breakdown during Krebs cycle. We can see that there are 6 NADH and 2 FADH and 2 direct ATP will be formed during Krebs cycle. That happen in the matrix of mitochondria. We can see that. And ETS, electron transport system or electron transport chain or terminal oxidation takes place in inner membrane of mitochondria. During this process, NADH, FADH completely break down and release ATP. That happened, that is called oxidative phosphorylation. Breakdown of NADH, FADH, release ATP. And last electron acceptor is oxygen. Oxygen. We can see that. Very important point, very important point. We can see that. Then respiratory balance sheet, efficiency of aerobic respiration is 45%. 45%. And amphibolic respiration is better considered to be as an amphibolic pathway is very important point. And RQ, what is RQ? CO2 by O2. That is maybe 1 or less than 1, more than 1, infinity or 0. Yes. These are the different points in this chapter. And we can see which are the different questions. Which statement is wrong for Krebs cycle? Which statement is wrong for Krebs cycle? Sorry. Which statement is wrong for Krebs cycle? What is Krebs cycle? We can see that. What is Krebs cycle? Yes. This is Krebs cycle. Very beautiful cycle, right? We can see that this is acetyl CoA with 2 carbon compound. There is condensation with oxaloacetic acid to form first stable product citric acid. That is 6 carbon compound, which is undergo dehydration to form isocitric acid. Isocitric acid undergo decarboxylation and dehydrogenation to form alpha ketoglutarate acid. 5 carbon compound formed during anaerobic respiration is alpha ketoglutarate acid, which is undergo decarboxylation, dehydrogenation, and to form succinyl coenzyme A, which is undergo splitting, produce succinic acid and produce GTP in the case of plants, in the case of animals, and ATP in the case of plants, and but GTP is immediately converted to ATP. And succinic acid undergo dehydrogenation and release proton electron accepted by become FADH2, and with help of enzyme succinate, dehydrogenase that is present in the inner membrane of mitochondria that participate in terminal oxidation and become fumaric acid is converted to malic acid, and malic acid undergo dehydrogenation to become oxaloacetic acid. And that is one cycle. Based on this question is there, which is wrong. There is one point in the cycle where FAD is reduced to FADH2. Is correct. FAD is reduced to FADH2 in this point only. In this point only, succinic acid, fumaric acid, in this point only. FAD is converted to FADH2. And next, during conversion of succinyl coenzyme A to succinic acid, a molecule of GTP synthesized. Yes, correct. GTP synthesized. Yes. Next, the cycle starts with condensation of acetyl group with pyruvic acid to yield citric acid. Is it pyruvic acid? No, it is oxaloacetic acid, a 4 carbon compound. The carbon number will be asked, the first product will be asked. It is also called TCA cycle, or it is called citric acid cycle, discovered by Krebs, therefore it is called Krebs cycle. We know that. Which is the primary acceptor? Oxaloacetic acid, not pyruvate. That is wrong. That is wrong. While you read the option Haribari, then you will feel the right. That may be give to, that maybe leads to mistake. Okay, and what about fourth? There are 3 points in the cycle where NAD is reduced to NADH, of course. Here 1 point, here another point, and here another point. One cycle, three. And 2 cycles required for the production of, for the breakdown of 2-acetyl coenzyme A. Therefore, require 2 cycles, therefore 6 NADH per glucose by Krebs cycle. This is correct, which is the wrong one. We can see that 3 is the wrong one. Okay. And what about the next question? Conversion of glucose to glucose 6-phosphate. The first irreversible reaction of glycolysis is catalyzed by. We know that glucose is first productive process is glucose is phosphorylated by using ATP, phosphorus of ATP, and become glucose 6-phosphate with the help of enzyme hexokinase. Is the enzyme which act on the glucose, which participate during respiration. If it is anaerobic and aerobic, because the first process of anaerobic and aerobic respiration is glycolysis. And common product for anaerobic and aerobic respiration, common compound is pyruvic acid. What's the carbon number? Pyruvic acid, 3 carbon. How many molecules from one glucose? 2 molecules of pyruvic acid. Glucose is phosphorylated to become glucose 6-phosphate, again isomerization to become fructose 6-phosphate, again phosphorylated to become fructose 1,6-bisphosphate, again that is undergo splitting with the help of aldolase to form triose phosphate. That is 3-PGL and DHAP, dihydroxyacetone phosphate, and isomerization become triose phosphate, that is 3-PGL, again dehydrogenation and phosphorylation become 1,3-bisphosphoglyceric acid, undergo dephosphorylation become 3-phosphoglyceric acid, undergo isomerization become 2-phosphoglyceric acid, undergo dehydration become with help of enolase enzyme, enolase to form phosphoenol pyruvate, PEP. Photosynthesis is the making process of glucose, but respiration is the breaking. While breaking glucose, we can see the product which is formed during photosynthesis. PEP is there, and PGA will be there. 3-PGA, that is 3-phosphoglyceric acid is there. And last process of glycolysis is dephosphorylation, where this phosphorus is added to ADP and ATP to form. This is substrate level phosphorylation. ATP formation in glycolysis and Krebs cycle, that is substrate level phosphorylation. And here ATP will be formed with the help of enzyme pyruvate kinase and to form pyruvic acid. We know that. What is the answer? What is the option? Hexokinase is correct answer. First one act upon is hexokinase is the correct answer. Not enolases during this stage. We can see that. And next one, phosphofructokinase. No. Next one, we can see that aldolase. No. That is during splitting. What's the answer for this question? One is the correct answer for this question. And we are also giving this IAT Neet, this integrated batch also we have. Okay, do use your opportunities. We are here with you with full support. And we can see that next question. What is the role of NAD plus in cellular respiration? We can see that while cell respiration carried, what happened, what is while the site where oxidation or breakdown or dehydrogenation takes place, released electrons and protons accepted by NAD, and NAD is reduced to NADH plus H plus. This is carrier. We know that. What's the answer? It's a nucleated source for ATP synthesis? No. It is a function as electron carrier, of course. This carrier of the electron, which is happen when dehydrogenation takes place or oxidation takes place, it is a function as an enzyme? No. And next, it is the final electron acceptor for anaerobic respiration. Who is the final electron acceptor for anaerobic respiration? There is, we can see that in the anaerobic respiration, we can see that their first process is glycolysis. Then in the case of alcohol fermentation, then acetaldehyde will be formed, which is undergo, yes, acetaldehyde undergo decarboxylation. First of all, pyruvic acid and pyruvic acid undergo decarboxylation to become acetaldehyde. This acetaldehyde undergo reduction to become alcohol, ethyl alcohol in the alcohol fermentation by using proton and electron of NADH, which is formed by glycolysis. Whereas in lactic acid fermentation, first process is glycolysis, form pyruvic acid, there is no decarboxylation, there is no acetaldehyde, directly reduction and to form pyruvic acid. We know that. What's the answer for this question? Is answer 2. We had seen that it is function as an electron carrier. Next question. Respiratory quotient value of tripalmitin is RQ is CO2 by O2? CO2 by O2? Yes. And we can see that in the case of tripalmitin, C51H98O6, very important, plus 145 oxygen gives 102 CO2 plus 98 water. That is, if consumed oxygen is greater than released carbon dioxide, then RQ will be less than 1. Here RQ is 0.7. If consumed oxygen is less than released carbon dioxide, then RQ will be greater than 1 in the case of organic acid. In the case of anaerobic respiration, RQ is infinity. Whereas in the case of this carbohydrate, that is glucose, is used as a respiratory substrate, we can see that RQ is 1, 6 CO2 plus 6 O2. That is 6 O2 will be used and 6 CO2 will be released. That is CO2, 6 by 6 is 1. That's the point. We can see that what's the option for this question? Here is 0.7 is the correct answer. Then 0.3, next we can see 0.09 and we can see 0.9. What's the correct answer? Is 0.7. Triple that is 0.7 is the correct answer for this question. And next chapter, last chapter of +1 botany, we can see that plant growth and development, very important chapter. We can see that while go through the previous questions, we can see that in 2008, that in 19 and 15, there are 2 questions, and 2017, 16, there are 1 question asked. Very important chapter. We can see that. What are the different points in this chapter? Whereas plant growth is indeterminate because of the presence of meristem. And we can see that measurable growth is measurable. And we can see that growth phases. That is, we can see that which are the different phases of growth, that is connected with anatomy. That is, we can see that different phases of growth, and that is connected with morphology also, right? And we can see that growth rate. So the growth rate, there's arithmetic, geometric growth. And we can see that differentiation, de-differentiation, and redifferentiation concept is also the very important. Yes, and next we can see that development, that is, which are the factors affecting development also, and that is in the internal factor, that is intercellular factor is, that is between the cell. We can see that PGR, plant growth regulator or phytohormone, which are they? Auxin, then gibberellin and cytokinin are the plant growth promoters, and we can see that abscisic acid is the plant growth inhibitor, and ethylene is mainly act as inhibitor, then promoter. That is in the NCERT. Who discovered and what is the function? Very important, you have to be studying. And which are the different question? And photoperiodism is also there. Photoperiodism, that is very important. W. Gardner and Allard. Photoperiod is a long day plant, short day plant, day neutral plant. And we can see that this critical photoperiod and phytochromes, important vanillization, low temperature treatment. Okay, that's important. Next, we can see that. What is the question? Fruit and leaf drop at early stage can be prevented by the application of, we can see that, ethylene. What about ethylene? Ethylene, that is root hair formation and petiole elongation, node elongation, deep water rice plant. And we can see that flowering in pineapple, synchronized fruit set in pineapple, and mango, flowering in mango, and female flowers in cucumber. That's function of ethylene. And artificial ethylene available in market is ethifone, thinning of cotton, walnut, cherry. Very important. Very important. And next we can see auxin function, apical dominance, prevent, promote, prevent falling of older leaves, flowers, fruits. That's question. And prevent falling of younger leaves, flowers, fruits. That is early stage prevent. Promote falling of older leaves, flowers also by auxin. Then auxin, we can say parthenocarpy in tomato. And also we can see that rooting in stem cutting, flowering in pineapple, xylem differentiation are the function, and cell division also there. Whatever written in the NCERT, make code for the preparation. Next, gibberellic acid. Gibberellin, that is, we can see that length, elongation of length, that is elongate and promote shape of apple. Axial length elongation, very important. Bolting, malting, that is delay senescence. Antagonist to gibberellic acid is abscisic acid. Important point. Next, cytokinin. Cytokinin, cell division, overcome apical dominance. The new leaves, new chloroplast, lateral shoot, adventitious shoot formation. We can see that, cytokinin. And we can see that, what's the point? What's the question? Fruit and leaf drop at early stage can be prevented by auxin. What's the correct answer for this question? 2 is the correct answer for this question. We can see what's the correct answer? 2, auxin. That is direct sentence of the NCERT. Study like that. Okay. And next question, we can see that. What is the site of perception of photoperiod necessary for induction of flowering plants? That is leaf. Leaf, not pulvinus. Pulvinus, that is option from morphology of flowering plants. That is swollen leaf base. Pulvinus, very important. Fabaceae. And shoot apex, no. Then leaves, yes, leaf is the photoreceptor. And whether is phytochrome is there, the PR become PFR and PFR become PR. Very important point. Very important point. We can see that. And then lateral bud. No. What is the correct answer? Leaves. The correct answer. Leaves. Which hormone will be produced? Flow region. Flow region is the When light received by the leaf, that phytochrom will be activated and produce fluorogen that move to the shoot apex, initiate flowering. That happened during photoperiodism. We know that. And what is the answer for this question? 3 is the correct answer. And we are going to discuss last and final question. Yes, it takes very long time for pineapple plants to produce flowers. Pineapple flower connected with auxin and ethylene. That is, we already discussed flowering in pineapple by auxin and ethylene. Which combination of hormone can be applied to artificially induce flowering in pineapple plants throughout the year to increase yield? This question is lengthy, but concept is simple. If you know the concept from the NCERT, thorough knowledge, you can understand what's the answer. Of course, is it gibberellin cytokinin? No. Is it gibberellin abscisic acid? No. Is it cytokinin abscisic acid? Of course not. Which is the correct answer? Auxin and ethylene. Okay, what's the answer? 4 is the correct answer. Okay, do your best. One day you will be someone's hope and someone's hero. All the best from Darsana Academy. God bless you.